Hi everyone, my name is Yılmaz Oğuzhan Demir. Welcome to my presentation for microfluidics class. Today I'm gonna talking about organ on a chip and their applications. So, first of all, let's start with the definition of microfluidics and why it is so important for organ on a chip applications. Microfluidics is the term used for the control and manipulation of micro-sized fluids. They control this process with the help of canals in micron diameter, which can vary. Microchannels are very small, but they occur in many application areas due to their large surface area and high mass transfers. Due to these advantages, such as the reaction speed of microfluidics, their easy use in separation and detection process, it has been desired to be used in organ on a chip. So, under the light of these informations, now we can talk about organ on a chip and the meaning of it. The aim of this application is to create organ structures that can understand and imitate human physiology and to be able to experiment on them comfortably. Cell culture needs to control inside and outside of the cell. With the help of cell biology and micromachining, the organ on a chip can adapt to the external environment and perform an approximate physiological simulation. At the same time, it is very important to control a few more parameters. These parameters are divided to four parts, and actually, it's key ingredients and essentials for the organ on a chip design parameters. The first one is fluid shear force. Microfluidics can manage nutrients and discharge waste through micropump perfusion to enabling dynamic culture cell. The dynamic environment in which cells are located is more comparable to in vivo conditions than static culture. In addition, fluid shear stress induces organ polarity. Importantly, organ on a chip exerts necessary physical pressure on the normal biological functions on endothelial cells by activating cell surface molecules and associated signaling cascades. The second one is concentration gradient. Micro-sized fluids migrate or be transferred from one place to another in a laminar flow. This allows the system to operate in more stable state. According to desired physiological application, the flow rate, the velocity of the fluids moving in the channels can be increased or decreased, and you can also change the geometrical design of the channels. The third one is the dynamic mechanical stress. As all you know, each of the organs creates a specific pressure within the body. Therefore, this pressure also affects the human skeleton and muscular systems, which is under mechanical stress. Elastic and porous membrane structures are used to simulate this pressure in an artificial environment. And the last but the most important parameter is cell patterning. The organization of the human body requires a complex and ordered arrangement of multiple cells to form a functional whole body interactions. Microfluidics control cell patterning for the construction of in vitro physiological models with complex geometries. Surface modifications, templates, and 3D printing contribute to cell patterning on the chip. The 3D printing method enables multi-scale cell patterning by permitting the formation of hydrogel scaffolds. These materials can prevent mechanical damage with complex channels. The advantage of 3D printing is to allow user-defined digital masks to provide versatility in cell patterns critical for in vitro reconstruction of the cellular microenvironments. Considering these criteria, different designs can be made for required organ structures. There are different structures in the body that have different functions. For example, while the lung provides an air exchange in the body, the heart pumps blood with the concentration and relaxation movement, and the kidneys help to provide waste. Many examples like this can be given. Such physiological distinctions prove to organ-on-a-chip practices are separated from each other and must be created with different ways of thinking. High-resolution live imaging and drug testing for pharmacological studies are possible using such microfluidic devices. Understanding specific organ disease models, drug and toxicity testing, and biomarker identifications are some major application areas. As in conclusion, organ-on-a-chip applications have gained great speed. However, it has broad problems that can be solved in the long term. The materials used in these applications are very expensive to produce and operate. For this reason, this poses a problem as researchers prefer affordable and reusable materials. Another disadvantage is that the most of the information is in the chips. If the researchers alter the complex operation of the chip while collecting the experimental results, the system may fail. It becomes difficult to interfere with the chip from the outside. In addition, many sensors are used to simulate and detect the working principle of organs. No system has yet been developed that can process the output information received from these sensors. So, this issue is open to explorations and improvements with advantages and disadvantages, but it will provide a lot of benefits in the long run. Thank you for listening to me. That's all I have to say about this topic. Have a nice semester and take care of yourself.